Hi, everybody. We'll just wait for a couple of minutes more to let people join in. Yes, Yep. Hello, everybody. Thanks a lot for joining us in. This is the fourth Lucy Cherbas Memorial Seminar, and we have a wonderful talk lined up by Dr. Chun Han. And So just to give you a brief background, we are the Drosophila Genomics Resource Center, and we have about 500,000 DNA molecules pertaining to Drosophila. And whenever we have bad weather like yesterday, we not only have to watch out for tornado warnings, but also look out for power failures because we have to come back here and see if all the reagents are in good shape or not. Um, and we have also about 300 odd different cell lines, and thanks in large part to Dr. Lucy Cherbas. And she was instrumental in setting up the center and running it for quite a while. So a brief introduction about Lucy. She did her graduate work at Harvard and moved to Cambridge for postdoc, moved back to Harvard, and then came here with her spouse, Peter, in 1985. She's one of the founding members of Drosophila Genomics Resource Center, and she did a phenomenal job of establishing the center and accumulating over 130 different cell lines and actually helping a lot of people manage their cell lines, publishing protocols, and also participating in a lot of collaborative efforts to improve the reagents and the information that we have about all these reagents. And in the spirit of her service to the community, we, as per our previous associate director's idea, establish this seminar series where we invite guests of this nature. Until now, we have had Dr. Simcox, Moore, and Dr. Yamamoto all not only do fantastic research, but also generate a ton of reagents for the community. And this is something I really enjoy about the fly community, where you have uh, very, very established researchers generating a lot of reagents for the community. And in keeping up with this spirit, we have today's speaker, uh, Dr. Chun Han. He's at Cornell, and Dr. Han did his bachelor's at Peking and then moved to Cincinnati for his graduate work. He worked on heparan sulfate proteoglycans and how they shape morphogen gradients in Drosophila. From there, he went to UCSF for his postdoc, working on dendrite morphogenesis. and eventually established his own lab at Cornell, where he's currently an associate professor. And looking at his CV, one of the things that strike is the number of image and video awards that him and his group have received over the years, and most recent one being at the recently concluded Drosophila annual meeting at San Diego. And apart from that, he has a plethora of grants from the NIH, and he has some wonderful publications. And today he's going to talk to us about oops, talk to us about CRISPR and OptoTrap genetic and optogenetic manipulations in Drosophila. As I understand, he would be telling us about some magic they do in their lab. So before I hand it over to him, just a few housekeeping um, uh, announcements. If you have any questions, please put them in the chat. And towards the end of the talk, we will deal with them and Dr. Han would answer them. And without much further ado, I will stop the share and let Chun take over.
I will share my screen. Just a second, set it up correctly. Okay. Okay. All right, everyone. Thank you for coming. I really want to thank Daniel and Arthur for inviting me. It's a great honor for me to give a talk at this seminar series. Lucy have done so much work for our flight community, and she made so much important contribution. And even though we also develop tools, our work is in no comparison to her accomplishment, but we hope the tools we develop also can benefit the flight community in some small way. So I would like to first briefly introduce my lab, and we are interested in the cell biology of the nervous system, focusing on two biological process. The first is a neuron-phagocyte interaction during neurodegeneration, and second is a mechanism of dendritic morphogenesis. But our lab also are very interested in developing novel genetics and cell biological tools in Drosophila. And today my talk will be focused on our tool development. So our motivation for developing Drosophila tools are to manipulate genes and proteins in flies. And over the years, we have worked on optogenetics and CRISPR. So I will mainly talk about 3 techniques we developed over the years. The first one is called CRISPR trim. It's designed for tissue-specific gene knockout. The second one is called MAGIC. It's designed for mosaic analysis through CRISPR-induced somatic recombination. And last one is called OptoTrap, is for light control of endogenous proteins in a tissue-specific manner. So let's first look at the CRISPR. As you know, the CRISPR-Cas9 system have drastically changed how we do genetics in fly. After the effectiveness of the system was initially demonstrated by the lab of Harrison, Wildinger, and O'Connor Giles, we were thinking, how can we use the tool better? So we explored different methods and eventually came up with the 2 methods that rely on 2 pathways, DNA repair pathways for the double-strand break induced by Cas9. The pathway called non-homologous unjoining is very efficient for generating indels. So based on this, we developed the CRISPR trim, which is for tissue-specific knockout. On the other hand, homology direct repair is very useful for generating precise genome editing, for example, for making knock-in. But it also can generate DNA crossover between homologous chromosomes. So based on this, we collaborated with the lab of Marina Wolfner to make this technique called MAGIC. It's for mosaic analysis. So first, let's look at a CRISPR trim. The idea is actually very simple. So for every gene we want to knock out, we generate 2 guide RNAs targeting the shared coding sequence. And then by supplying a tissue-specific Cas9, we can generate either two indels in the coding sequence or a large deletion, and this will lead to loss of function. So this idea is demonstrated in this image in which all the somatosensory neurons of Drosophila larva express membrane GFP and also a nuclear RP, but only in a single class 4D neurons, we can knock out GFP and leaving only the RFP visible. So once the establishment of this method, we have been using this approach throughout many studies, and we have a lot of success with it. But I want to acknowledge that besides us, there's many other labs are trying tissue-specific CRISPR, especially Philip Port and his colleagues. For example, they developed a ubiquitous guide RNA with US Cas9, US guide RNAs with US Cas9, and more recently, US Cas12a with guide RNA arrays. But in today's talk, I will only focus on ubiquitous guide RNA with a tissue-specific Cas9. So for the talk to be useful for the audience, I thought it would be a good idea to focus on the key factors I think is important for the application of CRISPR-Trim. And also, I want to introduce some recent progress we made to make the technique more flexible. Because CRISPR is a binary system, we should think about the guide RNA and also Cas9. For the guide RNA, I want to consider the guide RNA targeting sequence selection and the importance of guide RNA vector designs. And also we're going to talk about the necessity to test the guide RNA efficiency. But for Cas9, we found the expression timing of Cas9 is very important for the application. And also we noticed that Cas9 strains more often suffer from non-specific leaky activity, but we can manage this with anti-CRISPR. And lastly, we developed anti-CRISPR balancers, which can make a CRISPR-Trim much more flexible. So first, let's look at the guide RNA design. As we all know, the guide RNA target sequence is very important for determining the guide RNA efficiency. And fortunately, multiple labs have investigated how the guide RNA targeting sequence affect the guide RNA efficiency, and they came up with lots of very useful experimentally validated algorithm for picking the guide RNA targeting sequence. So we use most often the so-called CRISPR servers from the UC Santa Cruz for designing our guide RNA because they offer the efficiency scores from lots of algorithm. And it's very easy to pick the ones that are predicted to be efficient by multiple algorithms. And in terms of the targeting sequence, targeting locations, we always pick two guide RNAs targeting the shared common coding sequence of all isoforms, and with at least one of them targeting early part of the gene, and hoping to get a reading frame shift mutation in the early part of the gene. We also prefer the targeting sequence targeting conserved residues because even missense mutation will have a high chance to generate a loss of function. So in terms of the guide RNA efficiency, our most important conclusion is that the vector design for multiplex guide RNA matters. And so in this experiment, we compared several designs of dual guide RNAs to knock out a ubiquitous EGFP from Drosophila larva. And in these designs, they all encode one guide RNA for EGFP, identical with identical target sequence. But these designs differ in the number of promoters used, the tRNA used as a spacer, as well as a scaffold for guide RNA. Hopefully, you can see that the performance of these guide RNAs can differ dramatically, and only one of them, which is number 4 here, can completely remove the GAP. And this particular version contains the glutamine tRNA as a spacer and the 2.1 scaffold. And it's the most efficient one we found. But we also found that increasing the number of guide RNA have a negative impact on the efficiency. In this particular version, we add one more irrelevant guide RNA to the most efficient design. We found a small drop in the loss in the efficiency of a guide RNA. And interestingly, we found that the tRNA seemed to be detrimental in the germline. In this experiment, we counted the percentage of GIP positive offsprings from the parents of that contains a germline specific Cas9, a ubiquitous GIP, and a guide RNA against GIP. We found the guide RNA containing tRNA seemed to perform relatively poorly compared to other version. So if you are aiming for the highest efficient guide RNA for the germline, and the best design would be using dual guide RNAs using dual promoters. So to summarize this part, we found that in the somatic cells, the guide RNA scaffold 2.1 strongly boosts the mutagenetic efficiency, and the glutamine tRNA is a more effective spacer for multiplex guide RNA. But in the germline, we found that tRNA spacer slightly reduce the mutagenesis efficiency. And we have deposited our vectors to both antigen and DGRC. If you have a very important gene you want to study, and you only have a few of them, but you are unhappy with the existing publicly available guide RNAs, you may consider generating your own transgenic guide RNAs using these vectors. So as you can see that guide RNA efficiency is very important, but how do we know whether guide RNA is efficient or not? Fortunately, we found a convenient assay to determine the efficiency of a guide RNA. We call this assay Cas9 lethal assay. In this assay, we cross homozygous guide RNA males to homozygous actin Cas9 LIG4 mutant. And if the gene of interest is essential, then we expect to see the progeny all die at some stage because the actin Cas9 is supposed to generate a mutation in most of the somatic cells. However, if the gene of interest is non-essential, then the loss of function in the flies should not cause lethality, so we should see the female being viable. However, in males, because the LIG4 is on X chromosome, and the male should be hemizygous for LIG4, then the double-strand DNA break will not be efficiently repaired. And that should lead to lethality of the male in larval or pupal stage. So all these results will be true if the guide RNA is efficient. But if you see any result deviating from these results, then that suggests the guide RNA is not very efficient. So I highly recommend you to try out this assay before you use any transgenic guide RNA for your experiment. So we talked about the guide RNA. Now let's look at the Cas9. We found the Cas9 expression timing is very important. Imagine the gene of interest you want to study is expressed at some point during development. Depending the Cas9 you use, it could be expressed later than the gene of interest or earlier. So if the Cas9 expressed later, the gene of interest will generate product that can last a little bit, can last well to mask the loss of function phenotype. This is called perdurance. However, if Cas9 expressed earlier, then the gene of interest may not be expressed before, will be mutated before it have a chance to be expressed. That will lead to a more complete knockout. So this model predict that earlier Cas9 will have a stronger effect on the loss of function phenotype. So this is the case. And here is one example. The NSF genes and the SNAP genes are important for secretion. So loss of function should lead to defect in dendritic morphogenesis. When we generate the guide RNAs targeting all NSF genes and also all SNAP genes, and then when we use Cas9 expressed in post-mitotic neurons, we only saw very weak or no phenotype at all. However, when we use precursor Cas9, we saw much stronger dendrite reduction. And we found that the Cas9 expressed in the precursor cell stage is better than a tissue better than the enhancer-driven tissue-specific Cas9, which is in turn better than GAL4-driven tissue-specific Cas9 due to the expression timing difference. So the tissue-specific Cas9 lines are very important for CRISPR trim, but how do we make them for your desired tissue, right? We developed 3 strategies to do this. If you have a very good tissue-specific enhancer, you could generate the enhancer-driven Cas9 using our gateway cloning vector. Or if you have a GAL4 that expresses in the desired tissue at desired time, you could convert the GAL4 into Cas9 using the hack donor fly strange we developed. And but if you're unlucky, none of these options are available. But if you know a gene that expresses in the desired tissue at a good time, you could consider knocking the Cas9 into the locus through CRISPR. And we have developed optimized donor vector for this purpose as well. We donated all the flies and plasmid already. So next time you could possibly consider making your own Cas9 using one of these approach. And we're also happy to answer questions if you encounter any problems. Okay, so when we're using tissue-specific Cas9, we notice that the Cas9 often suffer from some leaky activity. The idea is showing here. So with the same OK371 enhancer trap, the GAL4 shows a very clean expression in motor neurons. However, the Cas9 also shows some leaky activity in the glia cells. So how can we deal with this, right? And we thought about using anti-CRISPR or ACR. They're essentially a collection of small proteins that can inhibit CRISPR activity. And to know which one works well in Drosophila, we selected 3 that's known to inhibit Cas9 activity. So we tested that out in the fly larva. This is the control animal in which consists of a Hedgehog Cas9 and a GIP-based Cas9 reporter. So we can see a lot of activity of Cas9 in the epidermal cells. When we added the ACRs, we found only the ACR284 is sufficient to inhibit the Cas9 activity. So how do we use ACR to improve the specificity of Cas9? So I did this. Let's say if we have a neuronal specific Cas9, but also have leaky activity in epidermal cells, we can generate a tissue epidermal specific ACR. And hopefully by combining this, we'll generate a more specific neuronal Cas9. And we found this idea indeed work. For example, this is a neuronal precursor Cas9, which shows activity in both neurons indicated by the yellow arrows, also in epidermal cells indicated by the blue arrowhead. When we add an epidermal ACR, we see much cleaner activity just in these neurons. And but I want to point it out that for this idea to work, the ACR have to be expressed at the same time as the Cas9 or expressed earlier, because once the Cas9 make a cut in the DNA, inhibiting its activity by ACR is no longer meaningful. So this idea is illustrated here. This is the epidermal precursor Cas9 that has lots of activity, but when we use epidermal ACR, it doesn't impact the activity. So in some cases, we found that it's beneficial to keep the Cas9 gathering in the same fly, because by this way, we can cross this fly to any other flies to generate biallelic mutations in the progeny in a tissue-specific manner. However, doing so can cause 2 kind of problems. First of all, if the gene of interest is essential, the flies contain the Cas9 and gathering can sometimes lead to lethality. The second, lots of Cas9 have leaky activity in the germline, and this can make mutations in the target sequence and disable the CRISPR in the progeny. But we can solve this problem by generating a balancer containing ACR. For example, if we add the ACR balancer to the fly containing Cas9 and a guide RNA, and the fly should be healthy. And also, the ACR balancer should be able to suppress acting Cas9 activity in the germline to keep the target sequence stable. So to generate the enhancer, generate the balancer containing ACR, we hopped ACR onto 6 balancers, 2 for each of the chromosome X, 2, and 3. So we tested them out with fly containing a germline-specific Cas9 with GIP-based Cas9 reporters. And the progeny of these flies should be GIP positive when the Cas9 is active. However, if the ACR is doing its job, now we should see the fly being GIP negative. We tested one of the balancer with two germline-specific Cas9, NOS-Cas9 and BAM-Cas9. Without ACR, these Cas9 lead to over 90% of progeny being GAP positive. However, the ACR balancer reduced the GAP positive progeny to 5% for NOS-Cas9 and 0 to BAM-Cas9. So they are very efficient. Then we compared, we also examined the rest of the balancers with NOS-Cas9, and they perform even better. So we think this kind of these flies would be very useful for genetic study because we can keep guide RNAs and Cas9 in the same fly happily and stably, and you can turn on CRISPR only when needed by crossing out the ACR balancer. So this idea is illustrated in this particular example. For example, the nematide gene is encode an enzyme required for NAD production. It's needed for cell to survive. And when you remove it from the neurons, the neuron degenerate. So you can imagine that a fly containing neuronal Cas9 and a guide RNA for nematide will be pretty unhappy. But adding a balancer containing ACR, the neuronal morphology look normal and the fly is pretty healthy. So that's about the CRISPR trim. I want to talk now about MAGIC. It stands for mosaic analysis by guide RNA-induced crossing over. The idea is like this. Imagine we have a cell that just after the DNA replication, we can use a CRISPR and to induce a double-strand break in one of the chromatid. And this can induce HDR DNA repair and leading to a crossover and a chromosome exchange between the homologous chromosomes. And in subsequent mitotic recombination, mitotic division, in the so-called G2X segregation, and we can generate two population cells that are homozygous and for the chromosome distal to the crossing over side. And these kind of cells are called twin spots. If one of the chromosome contains a mutation, then 50% of the twin spot cells would be homozygous mutant for the gene of interest. So based on this approach, ideas, we generated special guide RNA marker transgenes to label the mutant cells either positively or negatively. So these transgene will be inserted in somewhere onto a particular chromosome arm where it's expressed guide RNA to target the same chromosome arm near the centromere. And this will lead to the two kind of two cells in the twin spots. One expressing one have two copies of the wild type chromosome and one without the transgene, which would be the mutant. And we in this version of MAGIC we call it NMAGIC. We express a BFP in the transgene as well. So the wild type cell will be expressing more BFP, and the mutant cell will be dark. And here is the wing imaginary disk showing these kind of clones. And in another variant we call the PMAGIC, we express GAL80 as the marker. The GAL80 suppress GAL4 dependent fluorescent marker expression. So in the progeny, only the cells missing GAL80, which is the mutant homozygous mutant cells, will be labeled by the fluorescent marker. And here is a single neuron labeled using this approach. Using this method, we can study function of an essential gene. For example, syntaxin 5, Sec5, and Rep5, they are all genes controlling vesicle trafficking. Homozygous mutant for these genes are lethal. We can generate MAGIC clones for these genes in neurons and observe very strong dendritic reduction. In another example, we generate the homozygous mutant clones for E-cadherin in larval epidermal cells. The clones are labeled by in the magenta by RFP, and the junctions of the epidermal cells are labeled by alpha-catenin. And hopefully you can see that the mutant E-cadherin cells have lost the alpha-containing GAP. This is consistent with the role of E-cadherin in recruitment of alpha-containing. So I'm sure you have heard about FRT flippage-based mosaic analysis, right? So it has existed for a very long time. Why do we need MAGIC then? We think the MAGIC have some advantages. First of all, the FRT flippage method requires FRT site on every target chromosome. However, the MAGIC system does not have that kind of a requirement. It can works with any target chromosome. And second, the FRT in the FRT flippage system, the crossover site is fixed. But with MAGIC, the crossover site is very flexible, which is determined by the guide RNA targeting sequence. And because of this, it's difficult to apply the FRT flippage method to existing mutations if they don't have FRT on the chromosome. But it's much easier to apply MAGIC system to all existing mutations. So that's why the method has a great potential for genome-wide genetic screening. For a similar reason, the FRT flippage system cannot be used on wildly derived flies. It's not easy to use it in other species, but the MAGIC system can be used on wildly derived flies. It can be easily applied to other species. Here are some images of a wing expansion disc that carries homozygous clones of widely derived chromosomes. And here is another evangel disc carrying clones in interspecific hybrid animal. So using this approach, we can study cells from different species, how they interact with one another in the same animal. So, and when we only demonstrated the proof of principle of a MAGIC, but to make it a very useful approach for everyone to use, there are still works to do. So in the past few years, we have been focusing on the following 3 areas to improve this method. First, we optimize the guide RNA marker design for both P-MAGIC and N-MAGIC. Second, we build a guide RNA marker kit for the all chromosome arms. And lastly, we established a safer approach for generating clones. I will summarize the main findings for each part. First, to generate a higher frequency of clones, we introduced the more efficient guide RNA design that increased the clone efficiency several-fold. The second, to make the P-MAGIC clones brighter, we destabilize the GAL80 and also reduce the expression. And each of these changes increase the clone brightness. And lastly, in our previous N-MAGIC version, we use a nuclear BIP, which is not really good for showing the cell shape, especially for large cells like epidermal cells. So we changed the nuclear BIP to cytosolic BIP and also added the HA tag. Now we can see the boundaries of clone much better, and also we can visualize clone using antibody staining. And so with that improvement, we generate a genome-wide kit for MAGIC. And we generated 6 guide RNA for each chromosome arm, 3 for N-MAGIC and 3 for P-MAGIC. And the triangle here showing the insertion site of this transgene on the chromosome, and the red arrow shows where they target in the chromosome. And we found that these guide RNA markers have a different efficiency for inducing clones. So it's possible now for any user to pick a guide RNA that fits their need, right, with the desired frequency in their desired tissue. We deposit all these flies already to Bloomington. So next time we are thinking about the mosaic analysis, perhaps you could try these things out. And you may notice that some guide RNA seem to be very inefficient, right? For example, this 40E1 for 2L is much less efficient compared to 40D2. But sometimes, so you may think this is not very useful, right? But sometimes high frequency may not be desirable. For example, when we're using the 40D2 to generate clones in the Drosophila adult brain, we saw lots of clones, and it's very difficult to visualize the projection patterns of individual neurons. But when we're using the 40E1, we only sparsely label neurons, and now it's much easier to see the projection patterns of individual cells. And with the kit we generated, we show that MAGIC is compatible with a wide range of tissues, including developing proliferating tissues and terminally differentiated cells, and diploid and polyploid and stationary cell and mobile cells. And traditionally, there are some tissues that are difficult to analyze with the FRT Philippis mosaic analysis. And one example is the Drosophila neuromuscular junction. But with MAGIC, we can easily generate clones in the motor neurons and analyze the neuromuscular junction. This is a clone for BRP, and this one for V-GLUT. With antibody staining, we can see clearly the loss of this protein from these mutant clones. And also, MAGIC can be used for gene discovery. When combined with a vast number of existing mutations in the Drosophila genome, especially the deficiency library can be a very useful approach for gene discovery. Deficiency essentially is a chromosome with a large deletion. And there are hundreds and thousands of deficiency in Drosophila genome that tile the entire genome, right? And by generating homozygous clones for deficiency, we can ask whether the genes deleted in a particular deficiency are important for a particular biological process. For example, we can look for genes important for dendrite morphogenesis. And this is an example of deficiency that causes neuron to grow numerous shorter branches. By dissecting this large deficiency with a smaller deficiency or with mutation in individual genes, we can quickly narrow down to the genes that are important, responsible for the phenotype. So by this point, you're probably thinking MAGIC sounds really good, right? What's the catch? So we found in our use of MAGIC, sometimes the Cas9 can lead to ablation of cells, non-specifically. And this is an example image showing epidermal cell clones in larvae. And we found some cells are small and rounded, and they look abnormal. But the normal epidermal cell should look bigger and also polygonal shape. And we found these abnormal cells are not necessarily correlated with a clone. And similarly, we sometimes see ablation of neurons when we're using a precursor, neuronal precursor Cas9. We think this kind of ablation is related to chromosome aberrations generated by double-strand break caused by Cas9, because people have noticed similar things in other systems. So how to solve this problem? Because the chromosome aberration is associated with double-strand break, we thought about using NICs. NICs are mutant versions of Cas9 that can only cut one strand of DNA to induce a NIC. And there are two NICs that are named after the mutation they carry. So we compare these NICs to Cas9 in the epidermal cells. And this is the Cas9 image. We see a lot of abnormal cells. But with NICs, we don't see any of these, which is obvious from the quantification. But the most important question is, can NICs induce clones? And gladly, yes. So this is an image showing the Cas9 in epidermal, in winged mandibular disc, and this is the winged mandibular disc with NICs. And this shows that NICs can also efficiently generate clones. And we found different guide RNAs also show the same relative efficiency for both Cas9 and the NICs. We also wanted to understand how the guide RNA cutting pattern may affect the NICs-mediated clone induction. So we picked a neutral locus and generated several guide RNAs. This gene is not required for fly to survive, so the loss of function is not supposed to change the fly fitness. So we combine these guide RNAs, and then with NICs, we expect to see a single NIC or two NIC that are cutting on the same strand or opposite strand, or two NIC that are close or far away. And as a control, we check the Cas9, and all of the dual guide RNAs can efficiently generate clones, and they are, as expected, are more efficient than the single guides. But with the NICs, we only found one that works well, which cuts the same strand also far away. So when using nickets for magic, we have to pay attention to the guide RNA cutting patterns. We also want to make nickets more available for people to use in magic. So we generate the Hike donor flies for converting GAL4 into nickets, and also the gateway cloning vector for generating nickets. And so for the magic part, we basically developed an optimized guide RNA marker kit for the all chromosome arms. And we found the clones can be readily induced in most tissues, perhaps except for muscles. And we found that guide RNA markers with different clone efficiency, they can be used for different purposes. And the nickets are the safer choice for inducing clones, but they prefer specific guide RNA cutting patterns. Okay, now I want to switch gears, to talk about OptoTrap. This is a system designed to trap endogenous proteins by blue light. Our motivation for developing this tool is to inhibit protein by light. But why bother, right? Because we already have a lot of approach for loss of function study, including RNAi, CRISPR, mosaic analysis, and also even conditional knockdown of a protein. But all of these methods suffer from some common problem. And first of all, they all work on the same entire cell. But if a gene is required for the cell survival and also for dendrite morphogenesis, and it's difficult to just remove the protein function from a branch. Also, all of these methods work on the entire cell, entire life of the cell. And for neurons, they take multiple steps to grow their branches. It's very difficult to remove the gene function only in a specific step. So for this purpose, however, the optogenetics can deal with this problem, right? Because we can use light to inhibit a protein function just in part of the cell. We can also alter the temporal patterns of light stimulation to fine-tune the signal input. So optogenetics have much better potential for extracting mechanistic details. So for this purpose, we developed OptoTrap based on two light-sensitive protein modules. The first one is called a Cry2-olic, which is a monomer in the dark, but it forms a protein cluster under blue light. The second one is called a magnets. It's a pair of a small protein that exists as a monomer in the dark, but form a heterodimer under blue light. To make OptoTrap, we fuse the Cry2-olic with PMAC, 3 copies of PMAC, and fuse the NMAC with a pre-protein. So under blue light, the Cry2-olic will form this protein cluster, and then it will attract the NMAC into the protein cluster. If we fuse our protein of interest with a beta protein that interacts with the prey, our protein interest will be trapped in this aggregate as well. So we think this method can have potential to inhibit proteins whose function rely on to be at a specific subcellular localization. To make this system more versatile, we generated several other variants. Besides the regular PMAC, there's also a faster version, PMAC Fast2, which associated and dissociated match faster. And also, besides the single copy of NMAC with a prey, we generate a tandem dimer NMAC with a prey. And this version should give rise to even bigger protein complex because of cross-linking effect. We can also try different prey-bait pairs, and we generate, we use a nanobody as a prey to trap GIP tagged endogenous proteins. We also use GIP, split GIP fragments as a prey and a bait. Because GIP11 is small, we can tag endogenous protein with several copies of this protein without affecting the protein function. And by using GIP 1 to 10 as a prey, we can trap our endogenous protein into even larger protein aggregates because of a cross-linking effect. And we also deposit all of these flies to Bloomington. You're welcome to try them out. So we first demonstrate that this system can generate protein aggregates in epithelial cells and neurons. And this is in the first movie shows Drosophila larval epidermal cells. The blue light only illuminates the left part of the frame. And hopefully you can see that NMAG shows aggregates only in this part of the frame. And the second movie shows neurons. We found the dimer version of NMAG with a prey works best in neurons. In this movie, we illuminate the dendrite only in the middle part of the frame, and hopefully you can see that the NMAG form aggregates only in this part, but not outside. So the system is very local, very instant. And we measure the kinetics. For the regular version, the association half time is about a half second. And for the regular version, it's 1.5 seconds. And the system is also reversible. And the half time for dissociation is 2 to 8 minutes for the fast version, and it's about 1 hour for the regular version. And by using nanobody as a prey, we trapped endogenously tagged protein tagged with the GIP. And we test a bunch of proteins with different subcellular localization functions and the sizes, including actin binding proteins, microtubule motors, and mRNA binding proteins, cell junction proteins, and enzyme. In all these cases, the trap are expressed in the magenta cells. And the upper panel shows the GIP tagged endogenous proteins. We grow these animals in dark and expose them to light only briefly before the imaging. We can see that was sufficient to induce cluster formation in the trap expressing cells. And our intention of making this tool is really to study dendritic morphogenesis. And a good system to perturb is microtubule cytoskeleton, because microtubule has been shown to be important for dendritic morphogenesis. And especially the motor system, because motors are responsible for transporting the cargoes along microtubule. Fortunately enough for us, my friend Jill Wildinger has developed the GIP11 tagged kinesin in flies. So with the GIP 1 to 10 as a prey, we can trap kinesin. So we first demonstrated that the kinesin can be trapped by the system. And this is a movie showing the mitochondria, and which are labeled by Amicheri. And in this movie, we illuminated only the bottom half of the neurons. And then the upper part is still kept in dark. Hopefully you can see that in the movie, the mitochondria can move freely in the upper part, but you see very little movement of mitochondria in the illuminated region. And you can see better in this camera graph, in which the diagonal lines show the mobile mitochondria and the vertical line show the stationary mitochondria. And we found the system is also reversible. Even after prolonged trapping, mitochondria can return to regain their mobility after 20 minutes in the dark. So that's good, but what is the consequence of trapping kinesin in the neurons? So as a control, we found that OptoTrap overexpression alone would not affect neuronal morphology. And also expressing OptoTrap in kinesin GIP11 flies would not change the neural morphology as long as we keep the animal in the dark. But the animal of the same genotype, when they grow under the light, we see the neuron develop very small dendritic arbors and shows very strong dendritic reduction, which is consistent with the role of kinesin in dendritic morphogenesis. So this is nice, but the question we really want to ask is how kinesin temporarily regulate the arbor construction. So for that, we grow the animal in the dark initially and move them to the light at a different developmental point. And by that, we can inhibit kinesin for 1 day, 2 days, all the way to 5 days. And interestingly, we saw qualitatively different phenotype. And meanwhile, we'll also wonder how neuron can recover from the kinesin inhibition. For that purpose, we grow the animal in the dark first and then transfer them to light. And interestingly, they also generated qualitatively distinct phenotype. But importantly, we found the two data sets generate different phenotypes. Even though in this experiment, the total duration of kinesin inhibition are identical, suggesting that kinesin is doing different things at different stage. To further analyze, to understand this result, we analyze the result in three ways. The first is arbor size. The second is the growth of high order branches. And lastly, it's the spatial distribution of high order branches. I will talk about each one of them. The first is arbor size, which correlates more to the growth of a low order branch, growth of low order branches. This is the result from dark to light transition. We see with the prolonged inhibition, the arbor size becomes smaller and smaller. That suggests that the arbor can keep growing during the entire developmental stage. But in the light to dark experiment, we see that the last 2 days were not sufficient for the low order branch to regrow. That suggests that the first 3 days are the critical window for low order dendrite to grow. And it takes more, I think 3 days for the recovery to happen. And for the high order branch growth, we found that just the last 2 days of inhibition was sufficient to eliminate all high order branches. That suggests that the last 2 days are really the critical window for high order dendrite growth. And we found the last 2 days of recovery, last 1 day of recovery was not sufficient for the high order branch to grow. That suggests that it takes at least 2 days for the high order branch to recover. This is distinct from the 3 days we observed for low order dendrite to recover. So why there is such a difference? We think possibly different cargoes are needed for the low order and high order dendrite to grow, and the cargoes are transported at different speed. And lastly, we look at the radial distribution of high order branches. Kinesin mutant neurons have this characteristic phenotype called proximal shift, which means that the high order branches shift from the more distal dendrite to near the cell body. We found this proximal shift phenotype in 4 days inhibition group. However, the 5 days inhibition group shows a weaker phenotype than the 4 days, and they also show the weaker dendrite reduction. This suggests that the early activity of a kinesin the first day seemed to inhibit the dendrite growth somehow. And to further, this can be better seen in this plot, which shows a show analysis. The y-axis shows the relative distance from the cell body, which is at 0, and the widest part of the violin shows the peak density of dendrites, high order dendrites, and we can see the 4-day inhibition has a stronger phenotype than the 5-day inhibition. So we try to explore this a little bit more by playing with the maternal contribution. In our experiment, we use homozygous kinesin GIP flies as a mother and cross with the homozygous GIP males. So in the progeny, all of the kinesin molecules are GIP tagged and can be trapped. But if we use heterozygous kinesin GIP mothers, some wild-type proteins will be transmitted to the final embryos, final animals, even if the final the genotype are still homozygous. That's called a maternal contribution. So we look at these animals. We found that they indeed also have a stronger phenotype than the 5 days inhibition group. That suggests that the residual activity and early activity of a kinesin can somehow generate a stronger phenotype. So this is the OptoTrap is also the only method we know that can remove both maternal and zygotic protein activity in a tissue-specific manner. Okay, so with that, I would like to thank all the people who have done the work over the years. That include a lot of people, the current lab member as well as the previous lab members. I also like to thank our collaborators, especially Mariana Wolfner. In fact, the idea of magic was from her. Without her, we cannot have the magic system. I'd also like to thank our funding agency, especially ORIP from the NIH. Thank you for your attention. I'm happy to take any question.
Thank you, Chun. So if you have questions, please type it into the chat box and take your time. I'll ask maybe the first question. All right, so the data that you show with the kinesin trapping is really fascinating, and I was thinking, does it affect the higher order branches more seriously if you do it from the dark to light first? So it affects them more seriously, yet it takes shorter to regrow, right?
So just to make sure I understand you correctly, you're saying from the dark to light experiment, whether the high order branches affect more than low order branches, is that right?
Correct. Yes.
Yeah. So indeed, we see we see the inhibition more severe on the high order branches because the earlier days they grow in the dark, right? So we found that the earlier time was the important for the low order branch to grow, right? To expand the arbor. So in these cases, the kinesin are active. That's why the arbor can grow to the relative size, but the high order branches cannot really grow well.
All right.
So while we are waiting for questions, so what I wanted to ask was that your Nikkei's cutting pattern, what is the key things that to consider when you have a combination?
Yes, so we don't know truly the reason why the nicking patterns make a difference. We have some suspicions, but we don't truly know. So what we found out is that we want to use guidelines that are a little far apart, like maybe a 1 kb apart, and cutting on the same strand. So it seems like that works well for Nikkis.
And again, with your optogenetic trap, how deep into the tissue can you actually do this kind of analysis?
Yeah, very good question. And I think this is very important for people who want to use this approach. So we have been mostly focused on the cells, more close to the cells surface of the Drosophila larva, like sensory neurons and epidermal cells. But I also have collaborators who have a look at this in larval brain, and looks like it works really well. So that's a kind of encouraging result. Yeah.
We have one question from Prabhat Tiwari. Her question is, can kinesin aggregates travel away from the site of illumination?
Yeah, very good questions. So we have done such an experiment. What we did was try put light onto the neurons and then watch the kinesin movement. And for we gave them persistent light exposure for maybe several hours. Over time, we do not see any movement of this puncta. Rather, we see the puncta becomes brighter and more discrete. So it seems like they keep attracting all the free kinesin into this aggregate over time.
We have another question from Patrick Wu. The question is, does spatial inhibition affect only one part of the dendrite arbor?
Yeah, also, this is a very good question. Initially, this is the case. If you limit the blue light to only part of the dendritic arbor, you will see aggregate formation and also protein inhibition just in that area. But it doesn't last very long because of probably two reasons. One is that the system is, the proteins are diffusible. And there may be other protein diffused into this region and also out of this region. The second thing is that the Drosophila cuticle is quite reflective, and it scatters light quite well. So even though we try to limit the light illumination to a small spot, right? So and over time the light will be scattered to other parts, and the system is very sensitive, and that could affect the other regions of the animal.
Okay. So the next question is from Mirka Olirova. She has two parts to the question, I think. So the explanation for different efficiency of the tRNAs, that's the first question. And then the second one is whether the opto tools would work for imaginal disc tissues.
Very good questions. For the first part, for the first question, tRNAs, we test a bunch of tRNAs initially to see which one works best, and the glutamine tRNA stand out. Why this is the case, I don't truly know. I think it's probably related to the processing of tRNAs, because some tRNAs, needed to be processed in some cases, not as much for others. So the particular ones that we used for glutamine may be more efficient, more efficiently processed by the endogenous enzymes. And the second question, whether the OptoTrap can be used in imaginal discs, we haven't tried, but I think it's very feasible, probably quite feasible, because the love body wall is pretty transparent, right? So as long as you can make sure you can deliver enough light to the tissue, the system should be sensitive enough to generate the protein cluster.
I have one more question while waiting. Has anybody done it on an ex vivo then? On ex vivo tissues OptoTraps?
Good question. No, we haven't tried, but I think it probably would also work.
Okay.
It's a challenging, however, it's challenging to do all these dissections in the dark, however.
That's true. All right, I think we'll just give people another 30 seconds or so. If there are no more questions, I'll thank Dr. Chun Han one more time, and you can unmute yourself and say hi, or just wave, give a thumbs up. Anything works.
Thank you very much.
Oh, there's a raised hand. Wait.
Oh, okay.
Raised hand. It's a raised hand. Okay.
I think
Yeah, Toshi. But I think it was a reaction rather than maybe a reaction. Cool. Well, I think that should be all today. Thank you everyone for joining us, and thank you again, Chun Han, for giving the seminar. It was
thank you.
it was great.
Yeah. Bye-bye.
Thank you.